So first, I really would like to thank the organizers to invite me to this amazing symposium. And I was amazed by the exciting presentation and the posters in front of, like before mine. So today I'm going to talk about some imaging techniques that we developed to study gene organization in single cells. So one question that intrigued people in my lab most is how one genome gave rise to so many different cell types in the animal's body. And for each type of cells, not only they have very distinct looks, but they also have different functional components being expressed. And the secret actually lies inside the nucleus of a cell, where there is a group of proteins called the transcription factors. They dynamically engage with the DNA and dictate which gene get turned on and off in a cell type specific manner. And the gene regulation has been extensively studied in the past 2 decades by using cell population-based techniques, such as biochemistry and genomics. So based on these results, people already pieced together what's a static picture of gene regulation look like. However, before I start my lab, really few studies actually focused on to study the kinetics of these biochemical reactions actually happening inside of the live cells. So at that time, we start with a very simple question. So basically, how a transcription factor actually find its binding sites in live cells. So let's assume if you are a transcription factor, you actually face a very daunting task. So basically you have to navigate through the 3D nuclear environment and search through 6 billion base pairs of DNA and eventually land onto a tiny piece of target, and usually only consists of several DNA base pairs. So our question is how efficient this seeking needle in a haystack process is. So we collaborated with pioneer imaging tool builders on campus. So we developed a single molecule imaging based strategies, allow us to study the transcription factor protein binding, and also the transcription factor diffusion dynamics in single live cells. And based on these results, we believe that transcription factors search for the target through a try and error mechanism. So basically, they have to diffuse, hop from one piece of DNA to another until to a point it actually finds a specific binding sites. And later on, we extended this imaging method to study protein-protein interaction and also protein condensate formation dynamics in single cells. And so after we publish these strategies, and they get pretty good reception in the field. And here just a subset of works that done through collaboration with my lab and utilizing these strategies and to look at protein dynamics in different biological systems. So today I'm going to specifically and focus on talk about one imaging technique that we developed called 3D ATAC-PALM to study gene organization in single cells. So the gene organization in mammalian cells is a quite big challenge to start study because in the human cells, we roughly have 6 billion base pairs of DNA. So if we stretch it out, it's roughly 2 meters long. And the cell actually packaged this 2 meter long DNA into a 10 micron nucleus. So if we package the same density of DNA into an egg, it will involve a DNA strand around 6,000 Earth equivalents. So there is a tremendous of DNA density inside the nucleus and really prevent us to use any imaging techniques to study detailed DNA associated structures. And this is especially true because of the Abbe diffraction limit, which state no matter what imaging technique you use, the resolution is limited roughly about the half of the imaging wavelengths used. So if you translate this to light microscopy, which suggests that around 200 kilobase pairs of DNA are packaged per diffraction limit spot. So which means that any genes within 200 kilobase pairs, you couldn't tell them apart very well. I think this is due to this reason. In the past 2 decades, the genome organization field has been dominated by a genomic technique called chromatin conformation capture, high C, like 3C and the high C technique. So for those who are not familiar with this technique, I will briefly introduce it here. So to study gene organization and the first cross-link millions of cells and the fragment the chromatin to small pieces using the restriction enzyme. And then the use a proximity-based ligation to ligate this cross-linked fragment and coupled with high throughput sequencing. And based on this high throughput sequencing data, they can reconstruct a large scale interaction maps across the genome. The advantage of these techniques is that it have a rich sequence information, and it also have high resolution. However, the one key disadvantage of this technique is that it's hard to translate these 3C measured interaction frequencies to true physical proximities in the cells. So anyway, so let's summarize what the people. Let's summarize what the people found. Okay. So currently, what the people found is that this genome organization. So basically, the genome is organized to accessible A and the inaccessible B compartments. And these compartments get further partitioned into smaller topological associated domains. And within each tag, the interaction is protected. However, between the sites, between the neighboring, around the neighboring tags, the interaction is being suppressed. And at the heart of this model lies into key molecules called CDCF and Cohesin. And people propose that these CDCF and Cohesin organize chromatin to form a chromatin loops through the loop extrusion model. And specifically, the Cohesin ring can extrude the chromatin until to a point that this extrusion process is stopped at the CDCF boundary. And so our question is whether we can develop techniques allow us to specifically study and image those processes in single cells. And so we still definitely have hope because of my colleague, Eric Betzig. He developed this super resolution imaging technique as well as this lattice light sheet imaging technique. And specifically, if we combine both, we can actually achieve the XY localization precision about 25 nanometer, and the Z localization precision about 50 nanometer. And if you translate this into DNA terms, it's roughly about 200 base pairs of DNA per localization. So currently we have the resolution to study the gene organization at the single nucleosome level. So with this technique, we first developed a strategy allow us to systematically map where these transcription factor stable binding events occurs in the 3D space inside of the live cells. And what we found very interestingly is that these SOX2 transcription factors, these stable binding sites, they form these high density clusters inside the nucleus, what we call enhancer clusters. And these enhancer clusters, they are spatially segregated from the head chromatin, which suggests that they are labeling some accessible chromatin structures in the cell. And at that time, one of my dreams is really to image what's the DNA structures actually underlying these stable binding events. So I always keep this question in my mind, and eventually I realized we probably can take advantage of a very popular genomic technique called ATAC-PALM, developed in Howard Chang and William Greenleaf's lab at Stanford. And specifically, this technique uses a hyperactive transposes TIM5 to insert DNA oligos with high density into the accessible chromatin. And in the early days, they couple this ATAC with high throughput sequencing, allow them to map the accessible chromatin sites in the linear genome. So we think we probably can also use this for super resolution imaging. So I contact Howard, so we decide to join force and develop this 3D ATAC-PALM imaging strategy. And specifically here, we put a photoactivatable GF549 dye into the DNA oligo, and the TIM5 can insert this this photoactivatable dye with high density into the accessible chromatin. And then we can couple this with lattice light sheet imaging, and this allows us to image accessible chromatin structures in single cells with high resolution. And what we found is that these accessible chromatin sites, they are also form these clusters and mimicking these SOX2 enhancer clusters that were observed in single life cells. And these accessible chromatin clusters, they are also spatially segregated from the heterochromatin. And what we found is that these accessible chromatin clusters, they are actually correspond to very well with accessible chromatin domains in the linear genome, as we can demonstrate here by like a DNA fish and coupled with ATAC-PALM. So with this technique developed and validated, so in the next step, we really would like to use this imaging technique, specifically look at the accessible compartment and how the accessible compartments is being regulated by the CTCF and Cohesin. And to do so, we first generated oxygen-induced diagram system allow us to very rapidly degrade CTCF molecules in the cells. And we couple this depletion system with ATAC-PALM. So what we found is that if we deplete CTCF molecules in the cells, we can induce dramatic clustering of accessible chromatin in single cells. And here we quantify the clustering with the pair correlation function. So the only thing you need to know here is the higher the amplitude here, which means the higher the clustering is. So here just give you a zoom in single cell view. So what we found, if we deplete a CTCF from the cells, those clusters, it seems become much more compact. And so as a result, the accessible chromatin actually have higher densities in the cell. So, as I already kind of allude to, there are two possible mechanisms actually leads to this enhanced clustering. One is through compaction, and the other is through spatial mixing. So currently, our evidence point at the compaction. Specifically here, if we use DNA fish to measure the domains of this accessible chromatin, and what we find is that after CTCF depletion, this accessible chromatin start to shrink. And this correlates very well with high throughput genomic data, which suggests after CTCF depletion, there are much stronger interactions within the accessible chromatin. So we also collaborate with Bing Zhang's lab at MIT. So Bing is an expert at doing simulation on the polymer model. And so what Bing found is actually the loop protrusion model can explain the compaction of this accessible chromatin very well after CTCF depletion. And so basically, if we get rid of the CTCF molecules, the cohesin can extrude across the boundary. So it basically can extrude on top of each other, which eventually will lead to the compaction of the volume. And at that time, we also, because this is a very easy experiment to do, so we also asked Bing to simulate what happens after we deplete cohesin. So what Bing found, if we deplete the cohesin, this can induce dramatic expansion of this accessible chromatin. And at the same time, this loop intrusion model also predict that after cohesin loss, we should observe much less clustering of these accessible chromatin in single cells. So, so we decide to validate whether this prediction is true. So basically, we use the same strategy. We established a very rapid depletion system for the cohesin subunit called RAD21. And this allows us to deplete RAD21 within 6 hours. And we do the ATAC-PALM experiments. And what we found is that in complete opposite to the model predictions, when we deplete the cohesin, we observe much stronger clustering of accessible chromatin in single cells. And so here just give you a single cell view. Here is a Y-type cell, and here is after we deplete the cohesin molecules from the cell. And this accessible chromatin is start to mix in space and form this higher density and much more connected accessible chromatin structures in single cells. And here I just want
15 minutes.
Yeah, here I just want to keep a really long story short. And it's basically, currently we identified a tug-of-war relationship between cohesin and a BET scaffold protein called the BRD2. And so what happened here is we got rid of the cohesin molecules in the cells. The BRD2 mediated weak interactions will kick in and start to mix this accessible chromatin. So when we get rid of BRD2, we can actually reverse this effect. So currently, the lead author of these two papers, so Frank is starting his lab at the Cleveland Clinic. And another, I think, a very supporting story that done by Danny in collaboration with Jenny Filipinka-Swartz is that Danny and we study a transcription co-activator called YAP, and this YAP protein can form phase condensate very rapidly after hyperosmotic stress. So what we find is that these YAP condensates actually can induce a de novo formation of accessible chromatin domains in single cells. So which suggests that protein interactions do provide very essential roles to organize accessible chromatin structures. So currently we are collaborating with tool builders and to basically push this technique and not only be able to do that in the light microscopy level, and we also want to do it in EM, so we can generate large scale EM data sets for these ATAC-EM. So we want to collaborate with computational peoples here. And so we want to develop machine learning like algorithms allow us to characterize cell types in large EM like data sets. And another direction in collaboration with Harriet Hess lab here is we are also trying to explore and to do high density chromatin stain and generate high resolution EM data allow us to map the chromatin fibers in cells. And coupled this with genetic perturbation like systems, this would allow us to really look at what people like proposed in the past 2 decades, whether they are right or wrong. And with all that, I really would like to thank you for your attention and also thank all the people in my lab and also a lot of collaborators in my for my lab. And without their help, many of those experiments wouldn't be possible. Thank you.
Thank you, James. So with we probably have time for one question.
Yeah.
Okay, there's a chat. So Jay is asking, do you have a method to take the sequence information into the ATAC-seq imaging? Yeah,
if you can couple that with in situ sequencing, I think that would be really amazing. So you will have spatial information of each DNA elements. I think that's definitely the goal that we are going to work towards. But currently, we still don't have a really good way to do that. Yeah.
Great. Any idea whether it's possible to, for example, target searching for RNA? Have you thought about this?
I think I will leave that question to you.
Okay. All right. Thank you, James.